Welcome back to G Fun Facts Online. Today, we are stripping away the surface-level news to dive deep into a biological discovery that fundamentally changes our understanding of how viruses operate. If you think you know how an infection works, a virus floats in, hits a cell, and breaks in, you are about to have your reality shifted. In late 2025, a groundbreaking study emerging from a collaboration between the Peking University Health Science Center and the Harbin Veterinary Research Institute revealed a new structural paradigm of viral transmission. They call it the Migrion. This is not just a new virus strain, it is a new vehicle for viral delivery, a hybrid structure that combines the mechanics of a cell with the lethality of a pathogen. Over the next 20 minutes, we are going to deconstruct this discovery, exploring the biology of the migrosome, the mathematics of the Allee effect, and the terrifying implications of trace-mediated transmission. Let's begin with the context. For decades, virology has been dominated by the image of the lone virion. We picture a spiky, microscopic ball floating through the ether, playing a numbers game. It bumps into a cell, tries to unlock a door using a receptor, and slips inside. It is a model of solitary warfare. But nature is often far more ingenious and terrifying than our models suggest. The discovery of the Migrion proves that viruses have learned to hijack the very mechanisms our cells use to move, effectively turning our own biology into an armored personnel carrier for infection. To understand the Migrion, we first have to understand the stage upon which this drama plays out. We need to talk about cell migration. In 2014, researchers identified a cellular organelle called the migrosome. This was a massive finding in cell biology. When a cell in your body needs to move, imagine an immune cell chasing a bacterium or a skin cell healing a cut. It crawls. As it drags itself forward across the biological matrix of your tissues, it leaves behind long, sticky tethers. These are called retraction fibers. Picture a piece of gum stuck to the bottom of your shoe. As you step forward, the gum stretches into long, thin strands. Eventually, those strands snap. But in the cellular world, before those fibers snap, the cell pumps cellular debris, damaged proteins, and chemical signals into small bulges along these fibers. These bulges eventually pinch off and are left behind as the cell crawls away. These abandoned packages are migrasomes. For years, scientists thought migrasomes were just trash bags, biological breadcrumbs left behind by migrating cells to signal their path or dump waste. But in December 2025, researchers studying vesicular stomatitis virus, a cousin of the rabies virus, noticed something that should be happening. Infected cells were not just spewing out individual virus particles as expected. Instead, they were actively pumping viral RNA and viral surface proteins into these migrasomes. The virus had hijacked the disposal system. The result is the Migrion, a portmanteau of migrosome and virion. This structure is a large, host-derived vesicle, roughly the size of a small bacterium, packed to the brim with viral genetic material and studded with viral spikes. It is a monstrous hybrid. This is where the story shifts from biology to strategy. Why would a virus build a Migrion? What makes it superior to a standard virus particle? The answer lies in the mathematics of infection. In a traditional model, a single virion is a fragile thing. It is exposed to the immune system. If it manages to reach a cell, it injects a single copy, or perhaps a few copies, of its genome. The host cell has defense mechanisms, innate immune sensors that patrol for foreign DNA, or RNA. A single viral genome is often detected and destroyed before it can replicate. It is a race. Can the virus copy itself faster than the cell can shred it? Often, the cell wins. The Migrion changes the math entirely. Because it is a large vesicle derived from the host cell's own membrane, it acts as a Trojan horse. It looks like self. It smells like self. Inside this membrane, the virus packs a collective infectious unit. Instead of one genome, the Migrion delivers a massive payload, potentially dozens of copies of the viral blueprint, all at once. When a Migrion encounters a fresh cell, it enters and releases this massive bolus of viral RNA simultaneously. The host cell's defenses, designed to handle single intruders, are instantly swamped. Viral replication begins immediately and explosively. The lag time that usually gives the immune system a chance to react is gone. This phenomenon is known in biology as the Allee effect, where a population grows significantly faster when there are more individuals present to start with. The study published in Science Bulletin confirmed this experimentally. Cells infected by Migrions began churning out new viruses hours earlier than those infected by free virions. But speed is not the only advantage. The Migrion also breaks the rules of entry. Virology 101 teaches us that viruses are specific. The flu virus binds to sialic acid. SARS-CoV-2 binds to ACE2. This is called tissue tropism. It is why a respiratory virus usually stays in your lungs. Migrions, however, appear to cheat. Because the migrion is coated in the sticky, signaling-heavy membrane of a migrosome, it interacts with cells differently. The researchers found that migrions could be taken up by cells via macropinocytosis. This is a cellular process used to drink fluids or eat general debris. It does not rely strictly on specific receptor interactions like lock-and-key mechanisms. Once inside the cell's internal digestion chambers, the acidic environment triggers the viral proteins on the migrion's surface, causing the membrane to fuse and dumping the cargo into the cytoplasm. This implies that migrions might be able to infect cells that the free virus cannot. It suggests a mechanism for pan-tissue spread, allowing a virus to move from the lungs to the brain or the blood with terrifying ease. This isn't just theory. The researchers tested this in live animal models, and the results were sobering. Mice infected with migrion-encapsulated virus developed significantly more severe disease than those infected with the same amount of free virus. They saw neuroinvasion, with severe encephalitis developing much faster. The viral loads in the organs were astronomically higher. This leads us to a new concept in infectious disease: migration-dependent transmission. Until now, we thought about transmission in two ways: cell-free transmission, where a virus floats from cell A to cell B, and cell-to-cell transmission, where cell A touches cell B. Migrions add a third mode: trace-mediated transmission. Imagine an infected immune cell crawling through a tissue. As it moves, it leaves a physical trail of infectious migrions behind it, like landmines. Hours later, a healthy cell moves through the same area. It encounters the abandoned migrions, eats them via macropinocytosis, and becomes infected. This allows infection to spread temporally and spatially in a way we never accounted for. The infected cell doesn't need to be present for the new cell to get sick. The environment itself becomes infectious. Perhaps the most disturbing finding is the potential for heterologous loading. In the chaotic environment of a co-infected cell, a cell infected by two different viruses, the migrosome machinery doesn't discriminate. It scoops up whatever viral RNA is available. This means a single migrion could theoretically carry the genomes of both influenza and RSV, or VSV and another pathogen. This brings two different viruses into the same recipient cell simultaneously, dramatically increasing the chances of viral recombination, the process that creates new pandemic strains. This echoes other recent discoveries in the weird virology space, such as vampire viruses, but this is arguably more fundamental because it corrupts the structural architecture of the host cell itself. So what are the implications for human health? While the initial study focused on vesicular stomatitis virus, the implications are profound. This could explain phenomena like long COVID. If viruses are hiding in membrane-bound vesicles, sitting dormant in tissues as landmines left by migrating cells, they might evade antibody detection. Antibodies typically hunt for spiky proteins on free viruses. A migrion, cloaked in host membrane, might be stealthy enough to persist for months. It also has huge implications for cancer. Migrasomes were originally discovered in the context of cancer metastasis. Tumor cells are highly migratory. If a tumor cell is infected, it could be seeding the body with migrions as it spreads. Conversely, scientists are looking at this as a weapon. We could potentially engineer migrions to deliver tumor-killing payloads, using the cancer cell's own migration habits against it. Finally, this sheds light on neurotropic viruses like rabies. We have long wondered how rabies moves so efficiently, from a bite in the foot to the brain. The migrion mechanism, hitching a ride on the transport and migration of nerve or immune cells, could be the missing link. But with this discovery comes a new avenue for defense. The future of antiviral therapy might not just be about attacking the virus, but about targeting the vehicle. We can imagine a new class of drugs called migrosome inhibitors. If we can stop infected cells from forming migrasomes, or prevent the loading of viral RNA into them, we strip the virus of its heavy armor. We force it back into the lone wolf state, where it is fragile and vulnerable to our immune system. This discovery highlights the critical importance of the cytoskeleton, the cell's internal scaffolding. Drugs that stabilize the cytoskeleton might turn out to be potent antivirals. In conclusion, the discovery of the migrion is a humbling reminder of the complexity of the microcosm. Just when we thought we had mapped the battlefield, the enemy revealed a new technology. Migrions represent a sophisticated, efficient, and deadly evolution of viral strategy. They turn our own cellular biology against us, transforming the very act of cellular movement into a mechanism of disease. But we cannot fight what we cannot see. Now that we know migrions exist, we can hunt them, study them, and eventually dismantle them. The era of the migrion has dawned, but so has the era of defense against it. Thanks for listening to G Fun Facts Online.